And Allison was kind of pointing it out that those pedigree relationships have a large impact in terms of the accuracies and what's going on, and that's kind of what I'm going to be talking about today. I'm going to start off by talking a little bit about accuracy in general. We see lots and lots of genetic correlations flowing all over the place. I want to talk about why those are important and what sort of impact those have, leading into construction of a genomic enhanced EPD. And so Matt referred to these training populations, these validation populations, these target populations, how they're interrelated and the pedigree relationships with them, and then close up with some implications in there. Now we're taking a look at the accuracy of an EPD. It depends on several things. And basically boils down into the amount of data that we have on an animal. And it can come from several sources, either the trait measured on the animal directly. We take a look at traits measured on related animals. We're going to get high accuracy. It's going to be on the progeny. We could also take a look at correlated traits, which might be easier to measure. And then the new big thing, taking a look in terms of incorporating genomic information. And the primary way we take a look about doing that is with the use of molecular breeding values. And the amount that they're going to be able to contribute is going to deal with the correlation of these different sources of data with the true EPD, the true expected progeny difference. And try and illustrate that with this diagram here. What we're interested in predicting is this animal's true EPD. We have several sources of information that can be coming in. We could have the trait measured directly on that animal, and its contribution is going to be controlled by the amount of the heritability of that trait. We could also be measuring related individuals. That trait measured on that individual is going to be related to that individual's true EPD for that trait, also through the heritability, but we need to get back to the true EPD for the original individual we're interested in evaluating. And so we have to go through two steps. The heritability of the trait, and so if that's low, the contribution is going to be less. But also the pedigree relationship between those individuals, the more distantly they're related, the less they're going to contribute in there. We can also measure correlated traits on an individual. Part of it is going to be controlled by the heritability of that trait relating back to the true EPD for the correlated trait. And then it also needs to go through the genetic correlation between the trait that we're interested in and in the correlated trait. And so those more arrows that we have to go through, the weaker they are, is going to lessen in terms of the contribution. If those genetic correlation here is high, it's not going to have much of an effect. If the genetic correlation is low, even if the heritability over here is high, we're not going to get much of a contribution. At this point, we don't have any genomic information. As alluded to earlier, we can look at a molecular breeding value as just another trait measured on an individual. It differs from other correlated traits in that it should typically have a heritability close to 1. There's reasons why it might not, but usually if that heritability is less than 1, that indicates that there's a serious problem involved in there. So I think Allison referred to on her slide with the correlations, one thing she pointed out, all those heritabilities were close to 1 in there. But when we do an MBV, that's going to be related to the true MBV's EPD. With that heritability close to 1, these values should be essentially the same. And so the main thing that, in terms of getting through here, we have a 1 here. Then we have the genetic correlation between that molecular value, EPD, getting back to the true EPD, through that genetic correlation. As we make that path, we're kind of multiplying this times this correlation over here. That's about 1, so we usually don't talk about the heritability very much. But its contribution, and it's why you see in all the literature, is going to be that genetic correlation. It needs to go through that path. Okay. So in terms of that genetic correlation, the main one I'm going to be interested is in the heritability of that the genetic correlation of the molecular breeding value to the trait of interest. That heritability should be close to 1. And when we're going through and estimating those correlations, one of the things we check first to see whether or not we have a lot of faith in the results is we take a look at that heritability. If it's less than 1, that's usually diagnostic in terms of the fact that there's something we need to kind of go back and check into. And the reason one of the reasons we're interested in that genetic correlation is it's going to be related directly to that BIF accuracy. And we can take a look at what that BIF accuracy is going to be from that genetic correlation, assuming all we had was no phenotypic information, just that molecular breeding values. This is the maximum gain, and it's going to be a function. And I know Matt was worried that there wouldn't be any formulas, so I got a formula. It's just I was worried he wasn't going to put one in. Fancy, but the main thing in here is it involves that genetic correlation. The other thing you should notice, it doesn't just involve the genetic correlation. It's the genetic correlation squared. So in some of the talks later, you'll see a lot more focus in terms of the proportion of variation being explained. And that's because that its impact doesn't come in directly through the genetic correlation, but through its squared coefficient. That's really telling us what its contribution is. Okay? Now, when we're taking a look at genetic correlation, what we have is a measure of the association between the MBV, or that correlated trait, and the true EPD. Now, several things need to happen for it to have a strong association. And so we have these large SNP panels, single nucleotide polymorphisms, in there. And part of the reason for having a large panel is we're going to have a QTL, something that's actually controlling the trait. We want to have a SNP close to it. Well, if you have a lot of SNPs, it's a lot easier to get somebody close to it. So that's the first criterion. So you see a discussion, we want larger panels as opposed to smaller panels, assuming everything costs the same. The next thing is we have a SNP close to the QTL. It can't just be close to it. There has to be an association between the SNP alleles and the QTL alleles. There's a second test it needs to pass. And so we could have SNP alleles 1 and 2, QTL alleles, big Q and little q. And what we're interested is finding a SNP allele close to a QTL allele where there's a matchup between the 1 and the 2 and the big Q and the little q. It could be that the 1's paired with the big Q and the 2's paired with the little q, or it could be flipped the other way. Either way is just as useful as long as that there's a pairing where we find one of the SNP alleles paired with one of the Q alleles. But which direction it goes is going to be very important in terms of is it one with big Q or one with little q. And this shuffling is going to become important as we go through and we take a look as that pedigree relationship gets farther. These things aren't fixed. They can shuffle around in terms of those associations in there. The other thing is it's not just one QTL and finding one SNP. We have to have this happen many, many, many times. And so we want this to happen for many of the biologically significant QTL. The more often we can have it happen, the closer the association, the more that we have the pairing match up very tightly, the higher the genetic correlation, the larger the proportion of genetic variability explained. Okay? And so that's part of the challenge we face in terms of constructing these tests. Which leads me into the construction of a genomic enhanced EPD. And there's several steps that we're going to go through. It's going to involve the training data step, the validation step, and the evaluation step. And so the first thing, if we're going to have a genomic enhanced EPD, is we need to have a molecular breeding value, so that needs to be constructed. This comes from a set of training data, going through computer and people pulling their hair out as they try and get the programs to run and get everything cleaned up and everything to match up. And what comes out at the end is a set of estimated SNP effects which tell us, given a genotype, how to construct the molecular breeding value for that particular animal. And we can have different types of training data. We could have different types of models in which we build. Some might work better, some might work worse. But at this point, all we have is a way to construct a molecular breeding value. We don't know how well it works. In there? In there? So we're taking a look at that predicted SNP effect. Remember that it's going to be how tightly the SNP and QTL are linked. So if they're tightly linked, the more generations this is going to last, the farther that pedigree relationship can get apart and have it still work well. It's going to depend on that association between that SNP allele and QTL. It's not good enough to have something close. It has to have those particular alleles line up very tightly. And it's also going to depend on the magnitude of the QTL effect. If we do a really good job in terms of having a SNP match up to a QTL that's having an effect, if the QTL effect isn't very large, then it's not going to have a very big contribution. In doing this, what we're going to find is that when we build that training data set, it works really well with the training data. It's going to tend to be weighted towards animals with the most accurate phenotype-based EPDs. The more phenotypic information you have, the better you're able to detect it, and so it's going to work the best with those. Now, the other thing we want to bear in mind is we want to know how these MBVs perform not in highly accurate animals with highly accurate EPDs. You've seen the graph that Matt put up, and Allison talked about it also. The contribution and the gain that you make from having a molecular breeding value is not for animals with high accuracy. It's animals at low accuracy. So we're training, and in order to do the training is we need to have animals with high accuracy. These things are tuned to work with animals with high accuracy, but we need to bear in mind that that's not where we want to use it. These are designed to be able to be used for animals with low accuracy. And a variation of the set of graphs. I think you'll get tired of these graphs after a certain point in these talks. Here I have the graph, and this time I'm going to do it in terms of the jet correlation of 0.7. Matt had it in terms of the proportion of variation explained. This is explaining about 50%. As he pointed out, this is pretty good EPD, MBV. We get a lot of gain when we have very little information. As we go up and we have more and more accurate animals, the gain becomes less to a certain point where the contribution is fairly negligible. We're training up here. We're wanting to predict for the animals that are down there. Now, why is that jet correlation important? Well, let's say we said that the jet correlation is 0.7, but we're wrong. The actual genetic correlation is 0.5. I'm sorry, these molecular breeding values, when you calculate them, don't come with a label saying my true genetic correlation is 0.5. This is going to need to be estimated. And what you see in there, we think this is the accuracy we're getting. This is the actual accuracy that we're getting from that MBV. It also impacts that higher the genetic correlation, the more weight we're giving towards those molecular breeding values. If the value is too high, we're giving too much weight to those. So we want to make use of it, but we want to make proper use of it. And when we get the result out, we want to get an accuracy that accurately reflects how well it worked. Which leads us to the validation step. And what's going to be important here is we're going to have another set of data. It's going to be related to the training data in terms of pedigree relationship. We're not going to have a set of animals that are completely unrelated to those in there. We're going to run it through another set of software, another set of models. What's going to be another contribution is from that training data, we have those estimated SNP effects. That's one of the inputs in terms of when we're doing the validation. The thing that we're going to get out that's new is we're going to get an estimated genetic correlation. Now, at times, depending on the validation data we have, we might have really good estimates. Sometimes we have not quite so good, and we have those large standard errors because we put a lot of effort in terms of constructing this training data. Now we have an MBV. We need to go validate. This validation data has to have a lot of the same characteristics of that training data, and so getting that additional information isn't a trivial exercise. So the things we need to take to consider, okay, first of all, that genetic correlation is going to be expected to be smaller in the validation population. Remember, when we built that MBV, it was built to work really well with the training data. It's tuned because it worked really well in the training data. Nobody's going to be selling you an MBV if it didn't even work well with the training data. Okay? Part of the reason we expect it to be smaller is we're going to have a shuffling of those alleles. We could be really close in terms of having the SNP and the QTL allele close together. But those alleles, those 1s and Qs, get shuffled around. And the amount of shuffling depends on the strength of that pedigree relationship. Okay? And doing it, it has the same caveat that goes in that to do this, we need to have phenotypic information linked up to the genotypic information, and it's going to be, the biggest contribution is going to be coming from those with the most accurate EPDs. Okay? But we're not done yet. At a certain point, we actually want to make use of that information. Matt talked to us about the target population. There's going to be a third set of data, and at this point, we're actually making use of it in the animals we're interested in terms of those low accuracy animals. Okay? So there's going to be two sets of pedigree relationships that are important. We expect that correlation decreases as the pedigree relationship increases. Okay? Well, as we go to the validation population, there's a certain distance here. So that genetic correlation went down compared to what you get with the training data. The evaluation data is also going to be related to the training. And if this is a lot farther away, that genetic correlation that we're estimating at the one step is going to be too large and give us inflated accuracies. I guess it's possible that it's closer, but I haven't seen that discussed quite as often in there. So in building this, we're going to want to make sure that these pedigree relationships distances are about the same in there. And that's easy to say, a lot more difficult to do. If we take a look what's going into the evaluation data, we have stuff coming from the training data, those estimated SNP effects which are used to calculate the MBVs. Genetic parameters coming from the validation data in there. And we're getting our genomic enhanced or marker assisted EPD. And the big place, it doesn't matter if we mess it up for the high accuracy animals. We saw those graphs before, it doesn't make much difference. It's with those low accuracy animals where it becomes important. Okay? So if we take a look at what's going into those different data sets, that training data deals with the estimation of SNP effects. What do we need there? We need to have genotypes. It needs to have phenotypes. It needs to be phenotype heavy. If you're going to do a good job estimating, you need to have plenty of phenotypic information. As alluded before, that's why we start seeing things for growth traits and carcass traits a lot sooner than we see for disease traits and other traits that are harder to measure. Okay? And because it's built there, and not because somebody has a conspiracy, it's going to be tuned to work best in the training population because that's where we built it. The validation step, which involves the key thing in terms of estimating that genetic correlation, for that data set, we need to have genotypes again and phenotypes heavy. So it's very similar in that sense to training data, which makes it really tempting to say what we'll use is the training data for the validation step. The thing that we you need to bear in mind is that distance of that pedigree relationship, which keeps us from using the training data for the validation data. And that genetic correlation tells us how well it worked. When we get to the target population of the evaluation data, which is where we're actually aimed for, the phenotypes aren't as we don't have to have the phenotypes at that stage. We can only get so far without the phenotypes, but we can get part of the way. That we're going to see the biggest impact are going to be the animals with the least phenotypic information. And those MBV weights, how much do you weight it versus phenotypic information, and the accuracies you get out are going to be based on the estimated genetic correlation. The true weights and the true accuracies are going to be based on the true genetic correlation. Okay? And if we don't have that pedigree relationship correct, we're using the wrong value, even if we have a highly accurate estimate of the wrong value. So some of the implications. MBVs are built using training data. And as I said before, it's going to be tuned to work well with the training data. What we expect is that the genetic correlation, if somebody goes through and estimates that genetic correlation using their training data, which would be the first step that they would do, is where it's going to be the largest. As the pedigree relationship becomes greater between the populations, we expect to find weaker and weaker genetic correlations. Allison pointed that out. It's not surprising that as she moves farther away, that we see a general tendency for that genetic correlation to drop. Okay? And the thing we need to bear in mind is that validation population relationship to that discovery, the training that we're using, and the target. And the target are not the high accuracy animals going into the evaluation. It's going to be those low accuracy animals. Those need to be about the same. If it's closer, we're going to end up with too high a genetic correlation. We're going to end up with accuracies that are going to be higher than they truly are, extra weight given to MBVs, which implies less weight given to the phenotype than should be given. Okay. 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 Oh, I guess there's one more thing. And so if that genetic correlation is too high, then what we're going to expect is as the phenotypic information comes in, and after a certain while, if you keep collecting phenotypic information, even if you've got the accuracy, you have the genetic correlations wrong, it will eventually dominate that, and we're going to get greater changes in the EPD than we would expect in there. And I think that's it. Any questions?